A very good morning, good afternoon, good evening to all the participants who have joined us today for this 38th session, which happens to be a 2-hour online workshop on the analysis of genomes and microbiomes. We have an excellent speaker, a very young speaker today, Professor Lorena Carro, who's currently working at the University of Salamanca, Spain, as an associate professor. Lorena received her PhD from the University of Salamanca, working on endophytic actinobacteria in 2009. She has spent several years at the University of Lyon in France, working on the first steps of the interaction between Frankia and actinorhizial plants. And then she moved to the University of Newcastle to focus on the study of genomic information of actinobacteria and the application of this information to taxonomy. She's currently associate professor at the Department of Microbiology and Genetics at the University of Salamanca. Her main research areas currently study microbial ecology, genomics, and taxonomy. And she's been focused for years on plant-bacteria interactions and the diversity associated with those systems, including the soils, rhizosphere, and plant-associated microorganisms. Currently, she's studying the microorganisms from extreme environments and their application to fight climate change in agriculture. Editorially, she's been the associate editor of the BMSAB, the Burgess Manual of Systematics of Archaea and Bacteria, for about 4 years from 2019 to 23, and also for the Frontiers in Microbiology from 2015 to 17. And IJSEM, which happens to be the premier journal in systematics from 2018 to 23. Currently, she's also a member at large of the executive board of BISMiS, as I had already pointed out. So with this brief introduction, I welcome Lorena to start the proceedings and deliver her lecture first, and then walk us through the practical side of the analysis of genomes and microbiomes. Thank you, Lorena.
So thank you very much for this nice introduction. So first of all, I would like to thank to the organizer for allowing me here to speak a little bit about the things that we used to do in the lab and try to show other people what some of these basic experiments they can be also run on their research. So welcome to everybody. Good morning, good afternoon, and good evening, depending where you see us today. So we are going to talk about, a little bit generally how we can get the genomic information, which are the basic steps at the beginning, and then what kind of information we can get once we are able to generate a genome. So all is starting here, the beginning for most of us. We are going to work with cultivated strains. So we get something like that, then we try to get this. Then we get this isolate, purify them, make DNA extraction, and then we are going to try to identify the 16S RNA gene. As very well has shown last in the last seminar, Kamlesh also asked how we can work with this structure, how we can get and how we can purify this system. But this is one of the steps that we are going to use in taxonomy, the classification of microorganisms from the beginning of the polyphasic taxonomy has consisted in grouping many different analyses to very well characterize the strains that we are studying. So these include phylogenetic classification, several microscopical methodologies, biochemical analysis, and also these molecular methods. within the time, there are many publications that have shown us how there are some difference in this kind of methods, and from the phenotypic to the genotypic, which has been able to increase our accuracy and reproducibility of the data. This make us that each time we are going to rely more on the genotypic data. But we don't have to forget that the phenotypic analysis is very important, because with the genomic information, we have all the capacities that the organism has, but we need to really know if they are using or not. So this should be also characterized for a good analysis. So as it was shown before, everything for the sequencing beginning with the Sanger method. So we were able to start to determine which are the sequencing of, for example, specific genes of the 16S. But the method has some limitation. And when the scientific community confront the divide to get the whole human genome in the Human Genome Project, they have to start developing several strategies to be able to get all this huge amount of sequencing. They developed a different approach, including the hierarchical shotgun approach or the whole genome shotgun approach. And this was the base to develop what is called the next generation sequence. This next generation sequencing is we are going to be able to, when we have this high quality DNA, get into fragments. Normally prepare a library of these small fragments, and then make the amplification or cloning depending of the methods that we are going to use, by emulsion or by bridge. We are going to carry on many sequences in parallel. That this is going to allow us to get much more information much faster. This is what is called now the second generation for the sequencing, and or the next generation sequence. And as you know, we were developing many different machines with different characteristics, that some of them have survived better because sometimes for the accuracy, sometimes for the price. And there are some of them that we use as a routine now in the laboratory. Okay, for the last time, well, several years already now, it has been developed what is called the third generation, where we are going to include mainly the technologies of PacBio and Oxford Nanopore. These technologies, they are going to allow us a huge difference from the previous one, that is the obtaining the long read sequencing. And this is going to help us allow to close better of genomic information. We will see the importance of that a little bit. So for the last years, it has been increasingly sequencing many microorganisms, many bacteria, and their numbers are really amazing. We can check here just 10 years ago, we arrived to the 20,000 genomes of bacteria that were already obtained. But 10 years later now, we are over 2 million of microorganisms that has been sequencing, and that we have this information available. This is a lot of information that we can use for our experiments and analysis. Okay, the basis of all of this, as we have seen, is we got this high quality DNA from the purified organisms. In this case, when we are going to talk about microbiomes, we will use directly samples from the environment or other ones. Then we are going to divide this into these small fragments that we are able to get within this kind of sequencing, assemble them into context and generate this whole genome sequence. How we can do that is going to depend a lot of the capacities or laboratories. In some cases, many of us, we are going to rely in companies for generate this kind of sequencing. So what we can get from these companies, there are different elements. But one thing that I recommended all of you, if to ask them, in addition that of the assembled genome that they sometimes give us directly to us, to ask them to give us the raw data, the real reads from the machines that they have got. And why is that? Because we can work directly with this information. We can analyze by ourselves. And also because it's important to deposit this information in public databases when we are going to make our publication, to allow them and to liberate them for the community. Because we can, for example, generate different kinds of using different kind of machines. Within the years, the sequencing is evolved. So we can combine the data that we have generated for a company. Okay. And we can um, and we can combine them together and get better results. So I recommended you that because I have seen several times within the publication that when we can ask to the authors to include those data, they are not available anymore in the company that they have done this analysis. So it's more complicated. So ask for them at the very beginning. Okay, if we got them and we want to use them directly, first of the thing that we need to carry on is the analysis of the quality of those data. So we need to remove the adapters that we say before that we need to generate those sequencing, and we need also to evaluate the quality. There are different parameters that help us to understand if this read has good quality or bad quality. For example, we can use different programs like FastQC that we can use by command line, or there are another programs like Princeset that you have an online version and you can use introduce also your data there and see the different quality of the reads that has been generated. So depending on the quality of the reads, it's important to um, clean them. So we are going to trim our data. We can do that also with these programs normally, and we are going to maintain a Q quality of above 20 on this for all our data. So this is going to generate another group of data where we can only keep the high quality data. In this case, for example, in this example, you can see different kinds as well as here in the reads because these data are from Illumina, so we have, and they are paid. So we have the both directions, and this is why we see 1 and 2. Here is the same, but some are orphans, so they have no in the other direction, and it's classified different. So at this time, we will have this trimmed and curated reads. And this is the one that we are going to use to generate our alignment in different programs that generate the assembling of the data. And with the assembling of the data, that this is the ones that I told you that sometimes you got directly this file from the companies, is an assembly file in which you have already a FASTA document where is included all the context and all the basic information of the genome. So to generate this sampling, we have also several programs available that we can use. There are many, many of them. So I just give you some examples that you can use, but there are many others. Most and all of them are correct. We have not um, sometimes it's pretty difficult for the people to start on this topic to decide which is the one that I should use or which is work better. So there is no a correct answer. You need to try and see what programs works better with your data. For example, Spades is one of the most used, and Velvet as well. They work quite similar in the process of the data. They use bridging graphs. But for both of them, for example, you need to use command line, and you need to use to decide the cameras that you are going to use. Um, well, when we use with command, we just need to carry on our program, and we need to decide the characteristics that we want the program use, including these cameras. For the ones that are not associated with this terminology, what is a camera? I'm going to explain a little bit. And I think it's quite easy with this figure to understand the terminology. The cameras is the number of nucleotides that we are going to use to divide the reads that we have in our sequencing analysis. In the way that, for example, imagine that this is one of our reads that has 4 nucleotides. This sequencing could be divided into only one fork mer. You know, if we define that the cameras that we are going to use is 4, we only have 1. If we want to divide it in cameras of 3, we are going to have 2 cameras of 3, this one and this one. While if we want to decide 2, we are going to have 3 cameras, AT, TG, GG. Okay, so here you have another example with more reads with a little higher size. We can align them classically, or we can divide them in formers, each of one, and then make this analysis. This helps to improve the way that the sequencing are assembled between them to get this genome. When we have this assembly file, in both cases, if you have done by yourself or if you have got it from a company, in both cases, it's very important that you check all the information that is inside this document. Normally, you are going to have the basic information in the FASTA code, the cluster, the length of the cluster, and the coverage. We will see later also about the coverage. The coverage is going to give us a guarantee that this sequencing has been found several times within this parallel sequencing. And this is going to ensure us that this sequence is real enough in our genome. For example, you can see here some examples of sequencing that there really does not exist. This has been generated during sequencing. So we need to clean our data, both bases in the length. If we have very short sequences, normally they are not representative, and also in the coverage number. So if we have coverage very, very low, if we were able only to find one sequencing one time, probably it's not real. So pay attention to that before to continue analyzing your data. Once we have generated this file and this assembling, we have the genome that we can use to analyze our data. So we can use as well different programs available for carry on this analysis. QUAS is one of the most used of these programs, and we can compare several approaches. For example, here we have two. We have an analysis made with spiders or analysis made with fly. Fly is another program that used to make is normally used for sampling long reads obtained with PacBio or Nano. And we are going to have a series of different data that allow us to compare which one is the method that has working better. We can analyze how has been working this. For example, in the case as I said you before, if we use spiders with different number of cameras, we can see which assembling has worked better. And we are basing this decision in a series of characteristics. The program has also something that is quite nice because it's intuitive within the use of colors, because the red is indicated the worst data and the blue the best one. So directly it's more easy to compare, showing just the colors of our results. Within this data, we are going to have the total number of contexts that it has grouping. So when it's grouped in a small number of contexts, it's going to be the best. We are going to have the total size that we recover, the M50 and L50 characteristics. We are well, I just explained a little bit more about this characteristic, just to show you other programs to do the same, because similar to the other ones, was you need to use command line, but there are also some versions that you can use in directly with using the web based approach that is available to introduce your data, and you got visual results of them. Most of them, they are going to keep these main characteristics that we have talked about the size, number of contexts, etc., that we will see now. And so here just some names of different programs that you can use and test to evaluate the quality of your exam. This is what I call as important terminology. So the number of contexts is the total number of contexts that you have in the sampled genome. And why this number is important? Because the minus number of contexts that we have is the better, because that means that we have no spaces that we were not able to get the complete sequence or to be in between them. So there are some information that we can lose in this section. For most of the genome that you see now, well, now a little bit less because of the long reads, we are able to close some genomes, but if we are not able to close, there are some repetitive areas within the genome that makes it difficult to get only one complete sequencing assembly. So, but it will be the idea to have one context. Then the more number of contexts that we have, the less quality that has this analysis. Another important term is the full length. So this is the total size of the genome. And if we can compare, for example, with a reference genome, or if we know the usual number of bases that has been recovered in close genomes to the one that we are analyzing, we can determine if we have been able to recover most of the information or not from our sequencing. The following important term is the M50, that is that given a number of contigs is going to be defined as the sequence length of the shortest contig at 50% of the total assembly length. If you think like that, it could be complex, but I think it's pretty easy with this figure to understand. We have the whole genome here in the blue line, and we mark where we arrive to the 50% of the genome. And with this line, if we have put one after another all the contigs by size, when we arrive to the contig that cross this line, we can see that the M50 has this size. In this case, it will be 700,000. Okay, so this will be our value. As higher as is this value, for us is the better. So if we have that with the first contig, we arrive to cover half of the genome, it's going to be much more interesting. Okay, so it has been working better, our analysis. And the L50 is quite a similar value, but in this case, instead of check the size of the contig, we are going to check its position. So we are going to have the same, the half of the genome, and we say, okay, the one contig, two contig, three contig. So our L50 is going to be 3. And the last terminology that is interesting to remember is the coverage. So that is the number of times that a section of the genome is sequencing in a sequencing reaction. So this is what we talk about, these numbers that we can see in the here when we see the coverage. So this sequencing has been recovered 54 times, while this one has been recovered only 1.2 times. Okay, so for the coverage, as higher as we get the numbers, it's the better. Okay. Well, another way to verify how is our assembly and our whole genome is using other tools that allow us to identify the completeness of the genome sequence and the possible contamination in case they are not all the DNA that we have used it were sufficient, they were pure enough, and in this case that we can get another organism that has also been partially sequencing in our reaction. So for that we have CHIC-M. This is used also for both for genomes that has been obtained for single files, or it has been also so quite useful for the max that has been recovered for metagenomics or microbiome analysis, and with the beans it's more difficult to get these high quality values. So we are going to consider that over 95% is a good number for completeness, and under 50% for contamination are the values that we are going to work with. In addition to this program to analyze these characteristics, we have others. For example, Givoland is also a website where you can introduce your data and you also get information of the completeness of your sequence, and also some of the basic information that we have talked about before, M50, L50, or the percentage of GC, different information from your genome could be also analyzed. And this is for the case of the completeness. And also we have the CON-S16S. We will see both of them a little bit in the second part when I am going to show you how to introduce your data there. With the contest, we are going to be able to determine if there are contaminations. They are going to be based in the 16S to determine if they could belong to different microorganisms. And so this it will indicate that our sample or assembly is contaminated. Okay, well, this is the whole analysis that we can make to ensure that our genome sequence and our assembly is okay. So finally we have a complete, clean, with good numbers sequencing. And well, now what we are going to do with that. So I get used to get many papers in which the authors just generate all this information. They have down all this work just to use for a very small technique like the DNA hybridization in silico, or something just to confirm that it's a new species, and they don't do anything more with their genomes. So for me, this is really a pity. I think it's much more interesting to try to recover as much information as we can from the genome sequence and use that we have generated. So we are going to see some of the techniques that we can use, quite easy ones, to improve our analysis for the genome mining and understand which is the real information that we can get from those genomes. So the first things that we need to do with our genome and with our assembly, when it's ready, is to annotate it. So we need to make the annotation to identify where are the coding sequences, and to assume them in the case they have been previously analyzed to uh, different proteins or different um, genes, or even in the case we identify this coding sequence, but there is not a signal to something that is already known, as in them as hypothetical proteins. For this, we can use different platforms or pipelines. There is the NCBI pipeline available to generate the annotation. So we can download the information and carry on by yourself, but we can also use the NCBI website through the system of submission. We are going to submit our information that we can get some restriction times through the submission of the data, and they are going to carry on the annotation by ourselves. So the genomic information that we introduce in the database is going to put through the pipeline, and we get the annotation values for our assembly. Another one that we can use and is useful for the further analysis is the RAS system. You have this website that you can also introduce your genome there. We will see how to do that later. And you just need to register in this one to make your own account, but then it's free to use. So it's quite practical. We have also Prodigal that was developed by GGI, GMark, Glimmer, and many other ways for annotation that we can use depending on the characteristic of our genomic sequence. Some of the main elements that we are going to identify during annotation, they are going to be the ORFs, so all the genes, then the ones that correspond to the different kinds of RNAs, proteins, pseudogenes, or non-coding regions. And this will allow us to make further analysis of all this data. To compare with when we are carrying on this annotation phase, we need to compare with the databases. So for that, we also can use different kind of databases, including gene ontology, enzyme code, KEX, that is going to be used for pathways, Interpro, and many others. So we can use one of these pipelines that also includes some of these databases that works directly if we have not many idea of how to work or how to carry on this analysis or develop a pipeline by yourself. This is very practical. if we are able to develop it by ourselves, it's also interesting to know which kind of different databases that we can include in our analysis. So this is the application that I told you about, RAS for annotation. As I say, you need to log in here, okay, and then it has been also developed the seed viewer. Okay, in the seed viewer is going to allow us to see directly all the genomes that are publicly available, but also the ones that we have introduced and we have generated our own annotation by RAS in our account. Okay, so we will be able to see a lot of information that after this annotation process that we are going to assign many of these genes in different subsystems, and we will be able to access many of this information. We will see also later in the hands-on part of the workshop. So you see here also specific shares that we can do within the genomes if we are interested in some specific characteristics of our organs. Okay. So this is for general analysis and information, but we will be also interested in look for specific abilities. For example, the program Antiasmas through the website will also allow us to analyze, in this case, we are in the bacteria section, but there is also a version for fungi and a version for plants where we can look for the bioclusters, so the group of genes that are related with the production on some specific compounds, many of them related to with the secondary metabolism. So just we need to introduce our email, decide some of the characteristics, and introduce our genomic data, and we will get some of the results in normally few hours, one day, something like that. So when we got the results, we have got something like this, where we can see the different groups of clusters that has been generated. They will be we can classify them into the type, for example, for PKS, for MRPS, terpenes, siderophores, etc. And we have also lot of information of each of them. We can see the genes that has been detected in each biocluster, which is the similarity of these genes with the biocluster that has been already known or defined. We are going to have similarity values with the most similar known clusters. So we get a lot of information, and it's pretty easy to get. It's more time that we need to invest later for analyze them, but it's quite straightforward. So another one that we can use also to analyze our genome is going to be the GapMine system that is also available online. And in this case, we will need the protein sequence faster. We will see later how we can get this format file and just submit it directly to this website, and they will let us know if according to the genomic information we are going to have an organism that is able to use some specific carbon sugar. So it's going to be analyzed if they have the genes related with the degradation, the genes related with the acquisition, and according to this data, we are going to have different values of high confidence that our microorganism is going to use this compound, medium confidence, or low confidence. So this is also very helpful to have a general idea of the genomic information, and that we can also compare later with information that we got from our specific experiments. The next term that is also interesting for analyzing when we have a genome, mainly if you are working in taxonomy, are the different algorithms to determine how close our microorganism is to the other ones that are already known. So we can use this kind of analysis are the substitutes of the classical DNA hybridization that we used to carry on in the laboratories before, and now we have different methods that we can, different approach to compare this whole genomes information between them. For example, the ANI, average nucleotide identity, that is going to represent the average nucleotide identity of all orthologous genes that are shared between two genomes. This has a cutoff value considered of 94 at the very beginning that has been updated to 95, 96%. That is going to equivalent to the 70% in the classical DNA hybridization. What does mean? That means that if we got a value under 95% between two genomes, we can consider that they belong to different species if all the other analysis that we make agree with this, or if we have a higher value than 96, we can be pretty sure that these both organisms belongs to the same species. So from the classical ANI, there are some variation that has been developed later and we can use as well, like the ANI-B, ANI-M, or auto-ANI that we can find in different websites to carry on directly those different analyses to improve a little bit some of the limitations that have the original analysis. Another one that is highly used as well is what is called the digital DNA hybridization. In this case, that is based in the genome BLST distant phylogeny, is going to be generated an index between the genome that are evaluated. So they establish the regions that have high similarity between them, and then they apply a formula that is generated to determine this final value of similarity. So we can use it directly also online in the GGDC website. You can just introduce your genome and it's going to be compared with the other ones in the database, or you can compare directly between two genomes that you want to know if they belong to the same species or not. In this case, they have generated this algorithm in the way that the value that we got is going to be similar to the traditional DDH. That means that the cutoff is 70%. If we are over 70%, we can consider that both microorganisms belong to the same species. If we are under 70%, we are going to decide that they belong to different species. So one of the following things that we can do with our genome, and this is one that this is also applied in many of the manuscripts, is to generate the phylogeny with all the genomic information instead of use only the 16S gene. This can be done by two different approaches. One of them is the use of conserved core genes or the use of whole genome sequencing. Depending of what kind of analysis we are going to use, they have some variation of that. For example, the UBCG that was generated by the laboratory of Johnson, they have determined that there are 92 core genomes that are shared within the bacteria domain. So they have used these 92 core genes to generate a pipeline that they are going to determine if all of them are present in the sequences that we want to analyze, and then we are going to be able to make the concatenation of these genes and generate the phylogeny based on these 92 core genes. The executable files and the instruction manuals, you can find it directly in the FBioCloud website to carry on this analysis by yourself, or you can also use through the system that they have for users. Another website that allow us to use an analysis like this using conserved genes is the Microbiome analyzer, that is also a server that is going to look for group of orthologs. They're going to align them, and then it's going to generate a phylogeny. So we can use the system that is available online, and also we can do that but command line. In the case of the use of whole genome sequencing, we have also several options. As usual, we have many others. I just show you some of the ones that you can use, or that I found more easily to follow. But you have many others that you can also use. So in this case, with the RealFi, you are going to have the sequencing that are mapped through the reference genome, and then you are going to have the phylogeny inferred by Phi-Alp-M. And this program is also able to give SMP information in the case that you are interested on this kind of comparison. Another one that is classically used for many researchers is this website that has been developed as well for the DSM set, the Type-Strength Genome Server, in which you can directly submit your genome sequence. You can choose here the sequences that you want, but you can also have adding the genomes of the closest strains that you want to compare with, including if you want to include an outgroup, you can carry on that here. And then you can allow them to compare with other organs in the databases directly, or you can just decide to use only the ones that you have included. So you have many possibilities directly here online. You can include your email address and just submit your job. And this will allow you to get those kind of results once it's finished. So you are going to have a whole genome tree that you can download and you can visualize directly on the website. You have also the 16S tree. Similarly, you can download or visualize it. But you have also the digital DNA hybridization values that you can use. Normally for bacteria, it is recommended to use the formula D4, so to determine if it's they belong to the same species or not. And within the phylogenetic tree, you have the option to download in different type of formats, and this allow you to work with this phylogeny later. So that's mean that we can use, for example, iTools or other tools available to improve or change a little bit the tree that we got directly through these systems. So this is one of simple trees that we can generate with the ticks. But as I told you, with this, for example, if we go through the iTools, we are going to be able to generate quite more complex analysis and introduce many other information. Well, the last analysis that I want to show you is one that is quite complete one and very interesting for taxonomies. It was developed and published in 2021. and it's called protologer. Protologer make many of these analyses that we have talked about. So we need to introduce already the 16S RNA gene sequence. We're going to introduce the whole genome sequence, and then it's going to be analyzed, both of them. There is going to be evaluated for completeness and contamination. And then we are going to have a whole kind of analysis, including the taxonomic analysis with the different values and the corresponding phylogeny for both system 16S and whole genome sequence. We are going to have a ecology analysis of our genome data, and we are going to have also some functional analysis, including the carbohydrate degradation, different pathways, and antibiotic resistant analysis team. So this is quite complete and quite interesting to analyze. And you have in the own website a whole video to show you how to carry on each of the steps to introduce your data and to get all this analysis. So, well, here is just more detail about the kind of analysis that is going to be directly generated through this website. So a very quick example of what we can do with all this analysis is one of our works with micromonospora, in which we try to analyze what we can find under the DNA of these genus. So this work belongs to the JEVA project, so we carry on the analysis of many different genomes, about 42 that represent about 50% of the genomes of the group at that time, and we can classify them into within this kind of phylogeny using whole genome sequencing. We can classify them into 5 main groups. We can determine a relationship in them with some plant group characteristic. We determine their adaptation to several environments, how they resist in quite extreme conditions, and different abilities that they have also presented in their genomes, like latent enzymes, the synthesis of specific compounds like acetylene or trehalose, or PGPR characteristics that we can relate it all with the plants, and the capacity to produce a high number of bioclusters, because, for example, this genus is quite well known for producing antibiotics. So we analyze also the capacity of the genus, how we can determine the difference between the different species producing those compounds, and which were specifically related with some of these species within the genus. So I think there are many information that we can get from the genomes and that we should use it. So talking a little bit more specifically about microbiomes, I have just made some reference during the presentation, but the microbiome is going to be the community of microorganisms that exist in a particular environment. And we should be able to analyze all this information also for microbiomes. So the microbiomes are everywhere. The development of this next generation sequencing has allowed us to certify this, because before there were many environments that we were thought that they were sterile, but we are now being sure that they are not. They are the microbial communities over all the systems. And this development of the next generation sequencing and the analysis of the genomic information is going to allow us to give much more information directly from these environments without have to recover and be able to grow these bacteria. This is very interesting because there are many of the bacteria that or many of the microorganisms that they are not going to be recovered in the classical methods that we use in the laboratory, and this will allow us to study the whole community. We are going to have two main approaches. For one side is who is there, how we can determine which are the microorganisms that are in a environment, and for the other one, we will be determining what can they do. In the analysis of which microorganisms they are in a specific environment, we are going to have two main approaches that has been carried out for years. One is the amplicon sequencing, in which, for example, if we are working with bacteria, we are going to select the 16S gene, and we are going to amplify all the 16S genes within a community to try to identify who is there. But we can also amplify all the genome information and try to recover the genomes of each of these microorganisms in this community. So just to remember, this is a graphic that was also included by Kamlesh in the last workshop, but I just want to include it to show you one important thing, because many times when we check articles, we will see many of them talking about species or genus when we have carried out very short analysis of the amplicons. For example, the BC3, B4 sequencing, and yeah, even if we are able to sequencing the whole 16S using, for example, PacBio technologies, we will not be sure which species we have. We can be sure about the genus, but not about the species. So imagine when we only use this source section. For sometimes we cannot even sure that this information corresponds to a genus. So we need to be very careful when we analyze metagenomic data and the microbiomes in the use of the words and mainly at be sure at which taxonomic level we can analyze those data. So, well, this is the huge difference between the whole analysis of the 16S and the B3, B4 section. And just, well, I'm not going to be further on that because it will be another whole workshop, but just to let you know that when we use the whole analysis for the shotgun of a sample, we can have similar tools as we have seen for the genomic analysis. We have MetaSpaders, we have for the assembling, we have MetaQuast for the quality, we have MetaFly if we want to assembling long reads. And another important step when we use whole genome sequencing is what is called the reconstruction of each of these genomes. That this is one of the most complicated part of when we analyze the whole community. Because imagine if it was already difficult to reassign all these small parts to one genome, when we have hundreds of genomes that are divided in these small parts and mix it together, it's quite more complicated to assign properly. Each section belongs to each genome. But for this, it was developed the binding method that is going to classify this context and identify which could be more related and belongs to the same microorganism. So the pipelines, they are going to make an analysis of the sequencing quality. Then we are going to get the assembly and this binding, and then we'll analyze the functional and structural annotation of the genomes. Within the binding programs, I give you two of them, Concord or Metabat 2, that are highly used. For the second one, just to let you know a little bit how it works, we have several pre-analysis of the data, and then what this program do is to calculate the tetranucleotide frequency for each content and the abundance of each content. And with both data, they are going to generate a matrix of all this information that allow to identify different beings with the assigning the reads that correspond to each of them. At that point, we will have already a sequence that a genome sequence that it could be independently and it could be analyzed further with the other methods that we have seen before. Because if we have already a signaling that we have a recover a bean, that we have reconstruct a whole MAC, and it could be analyzed with the previous tools that we have seen before. There are also some proposals to improve the way that we can recover this complex and to improve the binding, for example, adding methyl groups to the DNA in the way that we can recover easily which ones belongs to each microorganism. And just to finalize this section, this first general information, and before to start with the second part, just to remember that to analyze what they can do in a community, we are going to use all the omics technologies. So for one side, we can understand what they can do. So the potential of the community, which genes are present, that for that we will use the whole genomic information that we have got, all the genes, and we will be able to determine if they have the capacity to do some analysis or not as a community. But to understand what is really happening in a community, what genes are really active and working, we will need to go to the other omics technologies, including transcriptomics, proteomics, or metabolomics, that will allow us a better understanding of what is really happening within a community. So we finish here the general information before we start a little bit with the hands-on analysis. So thanks a lot for your attention, and we can I can answer now some of the questions that you have generally before we pass to the second analysis.
Thank you, Lorena. This is awesome. We've got a lot of questions. Would you like to take them one by one, or shall I read the question and you can answer?
I'm going to try if I see them correctly and try to answer. If I lose
Okay.
something, please let me know.
Okay, no problem. Yeah, go ahead.
Okay, so I also recommend some manual best high quality genomic DNA extraction method for microbes in your species. Okay, well, which is a high topic because DNA DNA extraction is a key point always when we are working with this analysis. Well, for individual microorganisms, I think most of the commercial kits that are developed for genomic are working quite good now. Even for actinobacteria, that sometimes they are quite tricky on this. We used to get also quite quite high quality DNA that can be used for this. So this is not a big problem when you are working with a purified microorganism, but it is when we are working with communities. So if you want to use to get high quality DNA from environmental sample, for example, from soil, from plants, this is much more complicated. So you need to, depending of the sample, you need to check what has been done for by other people that are working with those kind of samples directly. What I can recommend in this aspect is also that well, I have no any benefits for that, but for example, for me it was quite well. The Kia hen kits they used to work quite well. What okay, what if we are not able to get a complete genome? This is not a problem. I mean, I imagine that you refer to when you get your analysis and you get many contexts. It depends of the number of contexts. Imagine if you have and also it depends of the size of your genome. The ideal, we have only one. We have more than 600, it will be too much. Why? Because we have genes that we need to be sure that our genes are complete when we are going to make the analysis. So if we have divided the genome into too many small species, it's going to be very difficult to be sure that we have the whole gene and the whole analysis that we are going to make after that is going to be limited to this. So in the case, um, but normally now most of the companies are given numbers under 200. I was just thinking in the very difficult microorganisms like yeah, the streptomyces that is quite huge and high GC. In the case if you have a gram negative or a smaller microorganism, it should be even lower. Yeah, below 100 or something like that. Can you give us an idea how different it is to assembly long reads data generated by nanopore sequencing as compared to Illumina MySeq? And we are going to use different set of bioinformatics pipeline. Thank you very much. Yeah. Well, the difference is there are already programs and systems to carry on those analyses. It is not that it's more difficult, but there are less options. What I mean is that most of the people has worked with the Illumina method, so there are many different options. It's very easy to find somebody that has the same problem as you before and how they solved it. And this is more difficult to find when you start to work with long reads. But there are pipelines to carry on that, and they also work. So I think they are going to be easily in the future because we are going to have more and more this kind of data, and the bioinformatics they are going to develop it more easy analysis for those data as well. If we are published a new species of microorganisms, can we just do whole genome comparative analysis of the bacteria together with its referent bacterial strains? What about the other chemotaxonomy? It is possible to publish a new species bacteria without this information. Okay, so well, what I mean is for me, it's important that the people remember that analyze of the microorganisms that we are work with when we want to describe a new species is important. It's still important to make as much experiments as we can to well characterize a microorganism. The genomic information is very interesting, and we should analyze them. We have them. And for example, it's very interesting to have this carbon source analysis directly from the genomic information. But I think it's still important to verify in the lab that that genes works and that they are really active. So we don't need to carry on everything but depending of the microorganism, we need to complete our analysis. We cannot base only on genomic information. For example, for me, if you are working with any kind of microorganism, I think for me it's quite important to know if they are an antibiotic that does not allow the growth, because this is information that could help everyone at any point. That when you characterize a new species, to understand if there are antibiotics that affect this species is something important to be known. For the chemotaxonomic characteristic, I think we rely in quite all technologies, and I don't think it should be repeated each time that we describe, for example, a species from a genus that is well known. Most of the genus, the chemotaxonomic characteristics are pretty well established and maintained. So for me, the chemotaxonomic analysis is interesting when you are going to describe, for example, a new genus, because you are not sure this is if they are going to change with respect to the closest one. So at that point, I see interesting. In a new species that normally all the species have a chemotaxonomic analysis very similar, it's not so reliable. And you can also carry on this some of these analysis using the genomic information. If I am interested in checking genes responsible for the capacity of the bacteria to degrade complex polysaccharides, I can check if in the assembly genome of my bacteria. So, for example, we will see a little bit about that in the second section. So, for example, using this the seed viewer, if you introduce your data and annotate with Rust, then we can go directly to seed, and we are going to be able to look for specific genes that you are interested and check if they are present or not in your bacteria. And also we can, once we have identified them and check that all microorganisms has it, we can go to the whole pathway and determine if it is complete or not, and how we can, and how it works. Okay. Are those of methods can be applied for seawater animals such as fish and fish? Um, well, uh, some they could be, but not all of them. If you realize in some of the programs that I have included, some of them are specifically defined for prokaryotes, but there are some of them that I have included just the name, I didn't focus, but they are designed for eukaryotes. So you can use them. They are normally they are specifically designed because of the different kind of information that we can have in the different type of genomes. But you have always a similar program to be used with those organisms. For bacterial microbiome analysis, it is safe to analyze the data for further statistical analysis using genus level identification of the bacteria. Uh, as I say, it depends of the analysis that you have done. I mean, if you are using amplicons and you are using a small section like B3, B4 or B5, B7, you cannot be sure, even at the genus level. You cannot talk. I know that we read a lot of manuscripts that talk in that way, but we cannot be sure of that. If you are using the whole, if you are working with shotgun information, or if you are working with the whole line 16s, yes, you can talk about the genus in the analysis of your data. Can we say that we have the same strain if we have any value of 100 or 99? No, not in. It's not the same. Well, probably if you have the 100% in any, it will be the same strength. It's something that I never think about before. But I'm not 100% sure that with those information you can confirm that it's exactly the same strain. Sometimes very close. In this I remember that they have some analysis in which we have very high value at the ANI, and when we have down the genomic profiles characteristic to identify closely were different. So they have some difference, some genes that are not exactly the same. and this is also difficult to be sure when with any, for example, you are not using the whole genes. So sometimes you can be avoiding the ones that are really the ones that make the difference between two strains. Okay, they are the same species, that you can be sure. What is the minimum value of coverage that we need for bacterial identification, especially if we intend to publish our findings? Okay. I will never work something below 10. Okay. At least that it has been amplified 10 times. But normally the average coverage, I don't remember, probably in the following workshop, Raul will be more specified with that, with those precise data. But I think it's not the same the coverage individually for each gene, okay, that we are analyzed in this whole file, that the average nuclear coverage of our genome. This should be higher. This should be working with 100 is okay in the average. What do we have to do to get the high quality DNA sequence? Um, we need to be quite sure. I mean, to get the high quality sequence, or I'm not sure if you are referring to the DNA extraction or later. But for the DNA extraction, as we say before, you need to be very sure before you sequence you send your sample for sequencing. You need to verify very well at that moment the quality. One thing that is very important, if we part from high quality um, DNA, all results they are going to be always better. And when we have the sequencing and we want to purify them, we always need to eliminate all those reads and information that has no the quality enough. For example, in QuAS, as I told you, that they are below 20 should be also eliminated because they are not good enough for the further analysis. What are the usual cutoff anti-SMART that is considered to have conditions? Okay, one thing with the anti-SMART that we have to be in mind is that the percentage that we got is the percentage of genes from the whole biocluster that are present. That not means that that the sequencing of the of each of these genes has this similarity. I don't know if I have expressed correctly, but if you check in the information of the original paper of Antias Mas, how it works, you have the percentage of the possibility of that the biocluster is the same because of each of the genes is distributed in the same way, and they have a percentage of similarity each of them. But I, for the anti-SMAT, I will, if you have 100% is clear. If you have 80%, probably is a kind of molecule that is similar to the one that is described, but it's going to be different. So you need to check also gene by gene, and you need to verify the real capacity of the microorganisms later to produce those compounds. But even with that, I think one thing that we should keep in mind when we use anti-SMAS is that um, it is um, it is very interesting, something that is not so high in percentage, in the way that if we have, for example, a PKS or an RPS compound, and is has some difference, we will be more lucky. I mean, we have more possibilities to get a different compound. So I think it's something to get into. We can give a general idea of what we find, but then we need to get into each of these compounds to be sure. Is there any recommendation of one site for analysis of comparative genomics or comparing or annotation of two or more genomes? Um, well, I didn't include here because it could take quite a long time. The GGI website in which we have a lot of information to compare between different genomes, all the information between them, and look for specific comparisons between your genome and others. But they have quite good, also available online seminars in which you can see how to do all those analyses. So that web, I think, is quite useful for that. My understanding of the max is that they are a kind of average genome, but they do not correspond to any precise individual. The 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 idea is that to recover a individual. It's true that the technology that we have now, it does not allow us to go to this specificity, and it's quite difficult. As we say, when we do the binding, it's quite difficult to split between high similar organisms, so sometimes we have several of them together. But this is going to be improved with the different methods, and it's time we are going to get more purified and more splitted organs. I think this is going to be little by little the future of this analysis. We are still in a kind of middle place in this way. How does this methyl group improve the genomic assembly? Okay, this methyl group, what it's going to do is just binding specifically to some parts of the DNA of some microorganisms. So then it's one of the things that we are going to take into account when we carry on the reconstruction. So the numbers of bindings of these methylation compounds is going to be another characteristic that will be included when we generate this general matrix of information to split them into groups. Okay, I think we have everything now.
Yeah, we covered all the questions. There were a few general ones that were very specific for the practical session. So I asked them to ask those questions when we come to that particular step during the practical session. Thank you, Lorena, for the excellent session. And don't log out, don't end the session. You can just, mute yourself, close your videos, and we will join back in 5 to 10 minutes.